Hello, my dear audience. Welcome to another episode of Let's Talk Micro. As always, I am so happy that you're here with me. Thank you for your support. And if this is your first time listening to the podcast, welcome. Okay, so let's go ahead and jump into today's episode, which is about Lariocidin, which is a potential antibiotic. And as you know, antimicrobial resistance (AMR), there are many challenges with it. And one of them is as we have a rise in these organisms that are very resistant to antibiotics, and sometimes we have this lack of available options, and we don't have a lot of new ones and a lot of new antibiotics being developed. So if I am reading an article about a potential antibiotic, I like to do an episode about it. So I hope you enjoy it. I hope you learn about it. So let's go ahead and listen to it. Well, as you know, in this podcast, from time to time, we do episodes about potential antibiotics, and we even do a series on like antimicrobial resistance, where we just discuss all the different classes and we shine a light on the problem of resistance. So when I'm reading some articles and I see a publication as something that's showing like a potential, antibiotic activity, I like to talk about it in the podcast. So today we're doing one of those episodes. So with me today, I have two guests that are going to discuss Lariocidin, and they are Dr. Manoj Jangra and Dr. Dmitrii Travin. Gentlemen, welcome to Let's Talk Micro.
Good afternoon.
Good afternoon, everyone.
Pleasure having you here. So for the audience, if maybe we can start with a quick introduction. I told them your name, so maybe what you do and anything else that you want to share with them.
My name is Manoj Jangra, and I am a postdoctoral fellow in the Wright Lab at McMaster University. So, in the Wright Lab, we are working on antibiotic discovery from the microorganisms. So, we collect like soil samples across the Canada and also from other parts of the world, and then we isolate microbes from this soil sample, and then we test if any of these microbes is making any new compound, or we call any new antibiotics, and then we characterize those compounds and then go on further.
My name is Dmitrii Travin. I'm a postdoc at Alexander Mencken Lab at UIC, University of Illinois at Chicago. And here we are studying ribosome and the biosynthesis of proteins in prokaryotes. And in part, we are interested in the compounds which inhibit protein synthesis. And given that the ribosome is a privileged target for the antibiotics, we are often contacted by the groups, including our collaborators from Canada, from McMaster, who come over some new compounds which are having antimicrobial activity and inhibit translation, and then we elucidate how these compounds are working, how do they bind to the ribosome, and the mechanism of action of these compounds.
Perfect. So welcome again. So let's start with some basics here, and I will post the link for the audience. If you want to read the article where they talk about Lariocidin and the whole process, you will have it there in the show notes. But let's start with some basics here. So what are the lasso peptides?
So, lasso peptides is a group of natural products which have been known for a long time. So it's not the first lysopeptide to be discovered, and we know them for decades. These are peptides which undergo post-translational modification, and they form a so-called Lariat form when a cycle is formed around the tail of the peptide threaded through the forming cycle, which give them a conspicuous um, lasso-like form, as if it is a lasso with its tail threaded through the cycle, the N-terminal cycle of the peptide. And lysopeptides have biological activities, although among more than 100 lysopeptides characterized to date, we know the mechanism of action of maybe 10 to 12 compounds. So we know the activity for around like 30, that they are antibacterial, but the exact mechanism of how these compounds are affecting bacterial growth is known for only a selected representatives of this class, and the majority of studied lysopeptide antibiotics are inhibitors of transcription. So they inhibit the biosynthesis of RNA, they inhibit the RNA polymerase. And recently, a couple of other lysopeptide antibiotics were studied in detail, and there were also found the inhibitors of protein degradation system, which inhibit CLP proteases, and some inhibitors of cell wall biosynthesis interacting with lipid 2. But these are just couple of compounds, and this gives us an idea that lysopeptides are not that straightforward as we would think of them one day as inhibitors of transcription. And the story we are going to talk today is falling within this paradigm just because we show yet another very different activity for lysopeptides. And this is all because of the form, because of this three-dimensional shape, so that they are fitting into so many binding sites on different enzymes on the ribosome, and we will talk about it later, I hope.
Excellent. So thank you for that. And so now let's start talking about as I was reading the article. So for the audience, so what is Lariocidin, and then there's also Lariocidin B. So maybe you can talk about each of those.
Yeah. So as Tima was talking about lysopeptides, so Lariocidin and Lariocidin B, so they are new members of this family. So Lariocidin is its 18 amino acid peptide, and as he mentioned that the initial 8 amino acid of this peptide, so they make a ring, and the remaining tail or the other end of this like peptide is threaded through this ring. So that's how we call them like glycopeptide. So Lariocidin B, it's the like variant or a very similar structure as the LAR, but the only difference is that Lariocidin B has another ring. So in Lariocidin, you just have one lasso, and in Lariocidin B, even the second end is tethered to the ring. So it's making a double knot structure. So it's highly stable structure, and they make the first class of peptides to act on the protein synthesis machinery or the ribosome we call.
We like to call Lariocidin be a pretzel peptide because it's a peptide which doesn't have either N terminus free or C terminus free, so both of the ends are linked to the cycle, which makes it like a small pretzel in a way.
Yeah, something like completely like entangled, so you cannot, no ends free.
Perfect. I love that. That's we always like visuals, right? And I'm sure that as the audience is listening to this, and great way to put it, pretzel. So thank you for that. Okay, so and then you talked about the mechanism of action. That's something that, of course, as microbiologists, when we're talking about our drugs, we like knowing and how do they work. Um, so how does this mechanism of action, how does it compare to other antibiotics that target the ribosome?
So ribosome is a target for multiple classes of compounds. So I can name maybe 20, 30 classes of compounds. Within each class, we have multiple. So a lot of different drugs are targeting the ribosome. So it's we sometimes when we say okay the target, we think of one enzyme, its active center, or something like this. With ribosome, it's the whole constellation of the binding sites and active centers because the activity of the ribosome can be inhibited in so many different ways because the reaction it catalyzes and the internal movements of the ribosome are so complex that almost each step can be somehow inhibited, stopped by small molecules, and we find more and more compounds which are binding to different binding sites. So if we think of the ribosome as a big molecular machine, there are two basically hot spots on the ribosome where the drug tend to bind. The one is in the large subunit of the ribosome around the peptidotransferase center. That's the place where the actual reaction happens, where the catalysis is happening. And many drugs are binding here, just to name a few. Chloramphenicol, nearby in the tunnel are the binding sites of macrolides, lincosamides, plurimutilins, whatever. It's a huge constellation of compounds. But today we are focusing on the small ribosome subunit and its so-called decoding center. This is the place where tRNAs and mRNA form the duplex and decoding happens. And another large group of the ribosome targeting drugs are binding around this area. And last time you were talking about the aminoglycosides, and it's a good example of compounds working in this area. And next to it, there are a couple of binding sites of different compounds. Many of them are not in clinics, but they, let's say, prevent the binding of the incoming tRNA. So the tRNA cannot reach its position correct. Also, very importantly, the small subunit is uh, moving during the translation cycle. So it has a head in the body, and the head is moving like this, especially after the accommodation of the A side tRNA. When the A side tRNA is coming, the head is moving to stabilize the duplex. And some of the compounds, let's say, can prevent this. So the exact mechanism of action can be very different. And for this, we should first to know how exactly the compound is binding to the ribosome and what does it change. For this, we have a couple of different in vitro and in vivo assays, which can allow us to understand what's happening and what's changed in the presence of the compound. And if we are talking about the mechanism of action of Lariocidin, we should first think of it as a pretty big molecule because Lariocidin, forming this three-dimensional shape, as we talked, this lasso thing, it's kind of a positively charged ball, a globe. Because of this positive charge, it can nicely interact with negatively charged ribosomal RNA. And it's binding with the head and the body of the small subunit and prevents the key step of translation, which is called translocation. So translocation is the movement of the ribosome to the next codon. So it does not prevent the binding of A site tRNA, but it does prevent the subsequent step, translocation. So it does not allow the movements of the small subunits which are required for translocation. It just sterically prevents the some level of it doesn't allow the movement of the 30th subunit to say. There are a couple of other compounds which work similarly. Not many of them are in clinics, but we can name a few also studied in our lab during the last decade. Let's say negamycin, adilarabdins. These are all compounds which were published and binding not exactly where lariocidin, but nearby, and also preventing translocation.
Excellent. Thank you for that great explanation. So and as you were introducing yourself, you mentioned that about growing organisms. So maybe if you can tell us a little bit about the whole process of discovering this compound, from growing. I know in the article it says like you are growing organisms for like a year, and that's to allow for some slow growing ones. So I'll let you explain it, but maybe if you can talk about the process of this discovering this compound.
Yeah, that's the fun part, I guess. So like usually like we isolate bacteria from like any soil sample and like it's nothing new. People have done that in the past. Also like other researchers in like other labs, they are doing similar kind of stuff. So what we changed this time, like instead of just isolating bacteria like within a couple weeks, we waited like for a year. And so why is that? Because if you isolate bacteria from like any soil sample per se, then the fast growing bacteria, they take up the nutrients and they grow like within let's say 1 or 2 weeks, and then we just discard the plates. But this time we waited so that even the slow growing bacteria, they can appear, and we did not use nutrient rich like petri dishes. So we used soil agar. So we used the nutrients from the soil, extracted them, and just put agar and just made the plates. So the nutrients are in the petri dish, they are from the soil. No extra nutrients were added. So it's kind of like their native habitat. We tried to mimic that. And then after a year, we isolated approximately 80 bacteria, and then we tested if they are making any new chemical substances or no. So I followed up on like several these bacteria, and then we ended up with this. We are calling it Bacillus species. So they are normal soil microorganisms, and they have been reported to produce many antibiotic compounds. Even this bacterium was producing a well-known antibiotic called colistin. So colistin is used to treat severe gram-negative infections in clinics. So the main idea that this strain is making something other than colistin, so that was the game changing for this project. So usually if you have a crude extract from like any bacteria, you test if there are any chemical substances or no. So we found colistin, but then instead of just like working on a crude extract, we segregated the extract in different chunks, so we call it pre-fractionation. And then we found one of the fraction was active against a multi-drug resistant strain of Acinetobacter baumannii, which is like a notorious pathogen, and also it's on the top list of the WHO's pathogens list. And this fraction did not have colistin. Now that was interesting. That time, the first time that I got to know that this bacterium is making something else, not just the colistin. And then I purified this compound from this mixture using different chromatographic techniques, and then we characterized and found it's a lasso peptide, and the amino acid sequence was completely new. And then our next question was how to figure out how this compound kills the bacteria. And then we collaborated with the researchers at UIC, and then finally we solved the exact mechanism how it kills the bacteria.
Excellent. And those of you in the audience, especially if you work with on the in the lab in microbiology, you're familiar with colistin, and definitely a very interesting history that it has. And I one time I had to do like a talk about some antibiotics and reading more about it, and how at one point in time it was kind of stopped being used, and then it was brought back because of the situation that we had with resistant organism and Acinetobacter. Great example that in the microbiology radar, that's one that's definitely out there with the with the carbapenem resistance and multiple drug resistance. So okay. So you mentioned with Acinetobacter. So for this, what organisms do you test it against?
Yeah, so once we like purified the enough quantity of this compound, then I tested again different like bacteria. So it's active against, you say, MRSA, the methicillin resistant Staphylococcus aureus, E. coli, then Klebsiella pneumoniae, and also mycobacteria. And recently we also found that it can also kill Pseudomonas aeruginosa too. So it's pretty broad spectrum, and it mainly attacks the aerobic pathogens. So it has minimum impact on the anaerobes. For example, our gut microbiota, we know they don't need oxygen. They are mainly anaerobic organisms. So we tested a bunch of the microbiota strains, and this compound had like minimum activity compared to the pathogens, which is good for us. It's like it won't harm your gut, it will only kill the pathogens.
Okay, yeah, and I'm happy you said that because that's always right a concern with coming up with a new antimicrobial that how does our normal microbiota will react against it. Is it gonna start depleting it, or so? That's definitely good to know, and that it show broad spectrum activity. um so other than you said, so you had no any other examples as far as observing any resistance. You said it targets aerobic bacteria from your study. So anything that you noticed maybe aerobic that it did not have activity against?
Manoj, maybe you can comment on some of the strains for which we have higher MIC. How, if, let's say, some of the gram negatives have high MIC, but we are currently working on it, especially Manoj improving it with modifications. And I can comment on some of the more basic ideas about the resistance to ribosome targeting drugs. And the first consideration, which people often miss, is that there are different ways how you can develop resistance to a particular antibiotics. And the first one is you modify your target. Let's say you have a mutation in ribosomal RNA, which does not allow the binding of the compound. And ribosome is, in the most cases, well protected against this because bacteria typically encode multiple RNA operands. So to get resistance through this mechanism, you need to mutate all of those, which is probably impossible. Let's say E. coli has 7 ribosome operands just to have more of the ribosomes. But for this, you need to have the mutations in each and every of those. and that's not the case usually. Second, you can modify your target. And for now, we do not know of any, let's say, methyltransferases that would uh, harm the activity, that would somehow influence the activity of Lariocidin, because there is one methylation site next to it. It's nucleotide 1207, but we have seen no effect of presence or the absence of methylation in disposition to Lariocidin, but any other methylations working in this area are not known. The next one, and for lasso peptides characterized to date, it's possibly uh, the bottleneck for resistance. It's uptake, because lasso peptides, they are peptides. They cannot go through the membranes just on their own. And for multiple characterized representatives of this group, this was shown that they are going through particular peptide uptake transporters. So they require special channels through which they enter the cell. And let's say mutation, premature stop codon, whatever in these genes would affect and render the bacteria completely resistant to these antibiotics. And this is not the case for Lariocidin. To date, we have not yet identified any transporter through which it may get into the cell, and we actually think that the uptake of Lariocidin is independent of the transporters. Maybe Manoj can tell you a bit more about it, because it's a very unique characteristic of the peptide, because it may explain the broad spectrum of activity. If you do not need a particular type of entry to the cell, you can enter through many cells, because the membranes are not that different as particular types of transporters in gram positives, gram negative, mycobacteria, and so on.
Yeah, thanks, Tima, for commenting on the ribosome part. So yes, Louis, as you said, resistance, that's the great question. So like we discover any new antibiotic, that's the first question we ask. Is there any resistance existing in the like clinical pathogens, or what are the chances that there will be resistance? So we have tested, so we have some E. coli strains that have resistance gene that corresponds to resistance of the clinical antibiotics. So we tested Lariocidin if there is any cross resistance or no. So far, we haven't found any resistance element in the clinical pathogens. So the only resistance that we can think of is in the producer strain that makes Lariocidin, and that's like only in the environmental bacteria. So there are less chance that the resistance can go in the clinical pathogens. And the second thing you asked about the aerobic versus anaerobic. So the only difference is why it's not targeting anaerobically as efficiently as the aerobic pathogens is the way it gets in the cell, in the bacteria. So most of the peptides that have intracellular target, so they use some kind of transporter proteins like Dima was talking about. But in this case, we could not find any transporter. Instead, this peptide uses the membrane potential. So we know bacterial membranes, even the like our human cell membranes, they have charge across their membrane. So we have the similar charge in mitochondria. And the bacteria, they like electric batteries. So their inside is like more negative versus like outside is more positive. So the cationic compounds, for example, like aminoglycosides or lyrosidine, so they use this membrane potential, this charge, and that's how they get in. And this thing is common among bacteria. So that's why it has broad spectrum activity. But in case of anaerobic bacteria, so they have less of this membrane potential because they cannot generate energy from like the oxygen. So instead they use like maybe fermentation or some other mechanism, so their charge is less. And that's why the lyrocidin, you need like more concentration of lyrocidin to have the same impact as you see in the aerobic bacteria.
Okay, excellent. And so when it comes to right, so
Louis, just let me add
please.
a couple of words about this intrinsic resistance of the producer, because I find it interesting. It's because when you're producing a drug, you should protect yourself, right? And this is actually not in the paper, which is out right now, but we are working on it, and it appears that the compound is modified within the cells of the producer if it is not exported out. So the producer has basically two lines of defense. The first one is we produce the compound and then we actively export it so that we can harm other bacteria around. However, if there is some disturbance in the regulation of this export and we have growing concentration of the drug, toxic drug, which would affect our ribosomes and our cell, we can protect ourselves with the second line of defense. And in the case of Lariocidin, this mechanism is acetylation. So this is the modification of one of the side chains within the drug, which makes the drug resist non-functional. Basically, it does not allow it to bind with the ribosome, and this is again one more mechanism how we can detoxify the compound. As I mentioned, import. Target modification, target mutation, direct modification of the drug, and also we haven't mentioned, but there are also protective mechanisms which can save the target. Let's say they can displace the drug with a specific protein from the ribosome. Tetracycline resistance has such mechanism, for instance. So the protection of the producer gives us an idea of what the resistance may look like, and is actually how we can make a drug resistant. A bug resistant to Lariocidin. However, as Manoj said, it's not for now only in soil bacteria. In this Penibacillus, we haven't found the homologs of this gene, which would likely acetylate Lariocidin in any of the pathogens just doing genome mining.
Excellent. Wow, it's very interesting, and I like how you mentioned, yeah, you use tetracycline as an example, so that helps the audience and get a little more visual and put it more in context. So as of right now, right, there's many steps from the moment that something is, you see something that's showing and um, antibacterial, antimicrobial activity, to the point that it actually eventually gets made and releases an antibiotic for people. So as of right now, what are the next steps for Lariocidin, if you can talk about it, or what are you doing next?
Definitely. So now that's the real challenge. So now we see the discovery, it was the easiest part. So the real hard work begins now. So we are trying to modify the peptide structure so we can improve its chemical properties, or so it can have better efficacy in animals, or like ultimately in humans. And then also we are scaling up the production, because bacteria, they make tiny quantity. They don't need they don't make drugs for us. So now we are working on like the strain engineering, the synthetic biology, to make this compound at a large scale, and also trying to modify structure so we have a more potent drug candidate. And that's the real like challenge. So we need a lot of resources, a lot of money, energy. So that's the hardest part now.
Right, definitely funding is always a challenge, and I like how you said, like they don't make antibiotics for us, right? It's just that's well said. Um, well, is there anything else that you want to add about this as the episode kind of comes to a close? Anything that you want to talk about regarding Lariciting or anything else?
Yeah, I can just mention like we didn't touch upon the toxicity thing. So we did some preliminary experiments in human cell lines, like the hemolysis experiment. So far, the lyrocidin had no cytotoxicity and no hemolytic activity, so which is great, because these cationic peptides, they like do not differentiate between the bacterial cell or like a human cell. If they are just have general mechanism of just interacting with the membrane, then they tend to be toxic. But in this case, lyrocidin has no cytotoxicity. And then we also did a preliminary animal infection model with this multi-drug resistant Acinetobacter baumannii. So we infected the mice in their thigh with this strain, and then we treated the mice with lyrocidin. And like in 24 hours, there was significant reduction in bacterial burden in thigh, in blood and spleen. And also all the treated mice, they survived, whereas the untreated mice, they died within like 24 to 28 hours. So we have a proof of concept that lyrocidin can treat severe infections.
And this, in part, requires this scaling up of the production because experiments with animals require much more of the drug compared to in vitro tests, structural biology, even work with MICs and bacteria, because you inject quite a lot of drugs, and for statistics, you need a lot. That so for all preclinics, you require pretty much.
Yeah. So you need grams or kilograms quantity that we are talking about for preclinical development. Now we can just make milligrams of it. So we are working on the next step now.
Yeah, and from my side, I would emphasize the evolutionary beauty of lasso peptides. So, in given that the modification of the peptide is the same in all of those, this is making this three-dimensional shape. For sure, in some lasso peptides, we have this some additional tailoring, so-called tailoring modification, but there is nothing major. But the whole concept of three-dimensionally constrained peptide, which evolution just fits into so many locations and so many functions. So it binds to RNA polymerase, it binds to the ribosome. Of course, it's not the same peptide. The sequence of the peptide is different, but the concept of the lasso peptide is and applicability of this type of a drug is underappreciated, I would say. And moreover, these compounds, which are encoded in the as the peptides in the genome, are a perfect thing for some genetic engineering applications. Because if we have the peptide initially encoded as its own gene in the genome, we can just modify amino acids. We can screen for the variants which are doing better, just making a libraries of these peptides and applying all the strengths of genomic engineering, genetic engineering to get better versions of these peptides. Unlike many small molecules, which cannot be that easily screened, you need to synthesize each and every of those rather than just having some high throughput technique, which is just based on the modifications, on changing the amino acids. And that's a strong side of these peptide based compounds and lysopeptides in part, in particular.
This has been so great, and I appreciate your time. And not only that, the time that you took to explain all the concepts, and I think we all benefit from this. It was like a nice way of all getting familiarized and learning. So I'm looking forward to see what's next, and it's always I'm reading publications, and if the door is definitely open, and if you wish to come back and talk more about this or any other thing that you're working on, you're more than welcome. So thank you for taking the time to be in Let's Talk Micro.
Thank you for inviting us.
Yeah. Thank you, Lee. Thanks for having us. It was so nice to talk to you about this project.
My pleasure. And that was the episode. So thank you so much to Manoj Jangra and Dmitrii Travin for joining the podcast and talking about Lariocidin. And I invite you to check out previous episode about potential antibiotics. There's one about lolomycin, there's one about fabimycin, and there's one about LPC 233C. And as you know, the Let's Talk Micro podcast is supported by the American Society for Microbiology. To learn more about microbes, visit asm.org. And if you are looking to learn more about antibiotics, please check out www.learnantibiotics.com and the Learn Antibiotics book available on Amazon. These resources include cheat sheets, practice tests, games, and more. And they are being used by thousands of people worldwide and may be helpful for you or your colleagues. So these are some great resources from Dr. Timothy Gauthier, a very passionate pharmacist. Okay, and I invite you to subscribe to the podcast. You can find it on all podcast platforms, like Apple, Amazon, Music, Spotify, Goodpod. And if you want to follow me on social media, I am on Instagram, TikTok, YouTube, and Blue Sky Social as Let's Talk Micro, on X as Let's Talk Micro 1, on LinkedIn as Luis Plaza, and I have an email address which is letstalkmicro@outlook.com. So please subscribe to the podcast, download episodes, and if the app allows you to do so, please go ahead and leave a review. I am so thankful for your support. And if you want to help the podcast in any way, you can do so via a Venmo link or via Buy Me a Coffee, KOFI. Both links available on the show notes. And thank you so much to those that have donated to the podcast. Your support is greatly appreciated. Thank you so much. And that, my dear audience, is the end of this episode. I hope you enjoy learning about this potential antibiotic, Lariocidin. As always, I enjoy sharing this information with you. Thank you so much for the support. Stay tuned. Great things coming your way. And hey, remember, are you going to ASM Microbe this year? If so, I'll be there. I'll mention more details about my event later on. But if you see me, say, hey, if you want a sticker, ask me. But I'm looking forward to meeting more people and connecting with all of you. So thank you so much. And please, continue bringing that passion to what you do. It's so important. You do such great work. So, as always, stay motivated, stay safe, and of course, continue talking micro. Until the next time. Bye.